Hello, hello. Are you audible? Hello, hello, hello. Good afternoon. Hello, hello. Are you audible? Hello. Right. Good afternoon, everybody. Today discuss about that sericulture, warm sericulture selection methods. How many types of selection? Uh, the processing of methods and how many types of selection methods. So those are divided by the four types of selection methods. The first one, individual and mass selections, and second one, fixation of characters, and the third one, formation of new breeds, and fourth one, authorization of new breeds. So this is the process of selection methods of sericulture. So the, this is the first important of objectives means the learn the importance of the selection methods, the understand about the individual and mass selections. Now about the methods of breeding, understanding about the formation of new breeds. So see the introduction. The silkworm germplasm in different countries of the world, and approximately information indicated that there are more than 4,000 silkworms germplasm accessories available in different countries. So germplasm is one of the breeding cultures breeding culture situation. So the more than countries, so ASEAN countries are like that to India, China, Japan, European countries also. 4,000 species are different countries. A very recent to completions of silkworm genetic stocks indicated that there are around 3,000 genotypes of Bombax mori. So what is the genotype? Genotype is the location of uh, the selection process of molecules. So this is the genotype. So for example, dimensional genotypes formula like that 4 is to 2 is to 1, 1 is to 1. Then so 3,000 around 3,000 genotypes of Bombax mori uh, at the global level, like that to Asia, China, Japan, and India. So global wise, the Bombax mori species or genotypes are 3,000 about genotypes are there. There which includes mutants, uh, parthenocleans, polyploid, geographical race. So complete genotypes are depend by the directly, indirectly affected to geographical conditions like that to temperature, humidity, and hair and hair transportation, gases transportation also. So both are directly, indirectly, directly affected to geographical risk. In fact, in fact, much much of the genetic diversity of Bombaxumor is derived from the inbred lines of land, race, and allied stocks. Allied stocks evolved by the silkworm breeds and also from hybridations of different geographical areas. So hybridations also depend by the geographical race because the geographical race important to growing of some like that species like that Bombax mori. Global levels are genotypes of 3,000 genotypes levels is there. So the hybridization of different geographical races is important because of this is the one of the selection methods. The mainly the Japanese, China, European, and tropical race, where is the districts of several economic characters. So this is the Asian countries uh, main important of Japanese, China, Europeans, and this is the three countries are tropical race. Like that, silkworms are temperature zone and a tropical zone, the growing of two particular geographical places. Like that, the first one tropical zone and the second one temperature zone. So tropical zone, some countries are, for example, Europe, Japan, and China. This is the tropical race. So then next one, how to the selection method, and how to control the natural and artificial. So the bivalent and univalent race produce high quantity of good quality silk. So I'm telling yesterday, bivalent and univalent is the uh, good qualities of production of cocoons and silk fibers, because this is the very important of fifth instar life cycle, increasing of internal organs also development like that, like the salivary glands. The salivary glands produce the silk fibers. Silk fiber, the fiber make the protein structure like that. Fibrin and sericin. Fibrin percent is the 74% and sericin percent is the 25%. So this belongs to protein molecule. So the good quality cocoons and good fiber threads are produced by the bivalent and univalent race. So then where is the multivalent race are RD tolerant to pathogen load and hereby resistant of disease compared to the bivalence and bivalent to produce low amount of poor quality silk. So multivalent is the one of the this production of silkworms, so multiworms, the anti disease resistance power, more than resistance powers is multivalent, because this is the incubation period is taken by the some days like that. So the silkworm, the silkworm lifespan is the very lowest lifespan. So because multivalent produce the silkworms, the more than resistance of disease. So thus those geographical races are very valuable genetic stocks for further important of silkworm race and the evolution of superior breeds of Bombax mori. Apart from a rich biodiversity of geographical races. So this scenario is very important of directly, indirectly impact to the growth of ecosystem. It is the because the biodiversity conditions is very standardization, because if it is the farmers are the growing of silkworms, depends by the uh, conditions of geographical conditions like that, to temperature, regulating of temperatures and regulating of oxygen transportation and regulating of humidity and regulating of bacterial disease and viral disease also. So this unique problem of global wise problems. So this multivalent race are very resistant of disease. Then there are also large number of mutants. The silkworm genetic stocks include more than 500 mutants for a variety of characterizes. For example, uh, cereal color, larval and adult integument colors, skin skin markings and body shapes, cocoon color. So those are how to find out the cocoons. So the visualization cocoons are mainly depend by the visualization. This is the color and shape, size, structure. So both are depend by the good quality cocoons. The characters is the marketing, integrating colors, skin markings and body shapes, cocoon colors and shapes, physiological strains, such as diaphases, vitality. The food habits and digestive, analyze blood and egg, esterase, alkaline and acid phosphatase, hemolymphoproteins, skin proteins. So both are lots are this is the physiological characterization and internal characters also. So those characters are deserved by the external features like that color, size, shape, and body mutants also. So some species animals are early mutants like that advanced mutant because of for example Bombax mori species of 3000 genotypes is there. So because one genotype structures is different and another genotype structures is different. So why? Because the mutants are very good growth to the specific location. The gene locus and the phenotypes were documented. Thus the geographical race mutants and the elatel breeds stock contains the major proteins of the parent of the silk germ plasm at the global level apart from the pathogenesis triploid, polyploid, and wild relatives of Bombax mori and Bombycidae. So complete global level apart, the germplasms are um, triploid, diploid, and polyploid. So what is the triploid and polyploid and diploid? The cells cultures to one cell to another cell. The mutants. It is called as it is called as uh, cell 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 culture like that. It is named as the fluorescence triploid, applied, and diploid. So the divided to cell germplasms and nucleus. Germplasms or nucleus are divided to the this based on applied and triploid and basis methods and basis. So complete this is the selection methods process to the hibernation to silkworms. Then how many types of individual and mass selection? So individual selections is the first one complete six types of individual methods. Egg color. Now this is the first important of egg color. The male and female egg colors are different colors and size and weight also different. The female egg is weight and size color are different to the male. The bluish color and bluish color, the weight is more than about to male. So male egg color is white color and very thin. This is the based on individual method. This is appear to the only visualization and very closely associated to the complete technical labor. The labors are choose the this is the particular individual methods. The next one egg colonial colors. So egg colonial colors is means the shape and size and structure. The colonial colors are different by the CaCO3. It is called as calcium carbonate. The calcium carbonate structurally and around to the different to the chronic colors. The next one, the generation egg generation to the next one larva generation next generation to uh, cocoon, next generation to adult stage. It is called as name as the butterfly. So the next two individual methods process is cocoon shape. So very important of cocoon shape. The good health and good conditions. Cocoons are good quality, producing the good quality of fiber threads. So this is the one of the individual methods of cocoon shapes. So the environmental ecosystems are defined by the cocoons are size, colors, and cocoon colors and size also. The cocoons are female cocoons color is a bluish color and size is the very large size and complete next generation go to the fifth instars stage is increasing of silk pipe threads. Then next one, individual methods are one of the next one. cocoon size. So cocoon size also very important because very good quality and good structures. Cocoons is size is very large size, and the cocoons are biochemical biological performance in cocoons like that gases transportation because the internal insters are internal insters are complete the uh, complete to another generation formation to another generations. So next one individual methods. So next one the cocoon nature and grains. So the cocoon nature and grains, the cocoon nature and grains completely very selective and associate to the technical peoples are grainage. So how many types of grainage? The grainage is structure defined by the shed structures and frame the on complete nutrients like that. So this is the cocoon nature structures grains. Then complete seven individual methods are appear into visualization methods. So first one egg color, egg chronic color, larva marking makings and cocoon shape, cocoon color, cocoon size, and cocoon nature of grains. Cocoon nature of grains. So complete depend by the individual methods. So next one, individual methods are quantitative characters. The quantitative characters are very important because uh, good quality and cocoons are produced by the special genomic and genotypes of populations. So because those are important to qualitative and quantitative methods also. This is process. This is qualitative quantitative also visualization process. So next one, the qualitative methods characters. So the how to the quantitative the percentage. Um, ability to in percentage ability means the habitats of uh, habitats of some special species to the some special species and convention to the special characterization to this particular race and so mostly depend by the habitats of very important of growing of silkworms. Why? Because this is the directly indirectly geographical status of depend by the this particular race and the particular production of the some importance. So So the next one, the uh, quantitative characters are depend by five types. The first one, ability in percentage. How many types of ability in percentage like that? How to calculation the abilities? Ability means is the growing of cocoons and growing of caterpillar structure and size. The internal structures also the ability. The ability means the complete species of lifespan depend by the lifespan also. So next one, larval duration in hours. Larval duration is in hours. So the silkworm lifespan is a very little bit timings. So the first instar development to 2 to 3 hours only. Then second instar 3 to 4 hours. Then third instar 3 to 4 hours. Then fourth instar 4 to 5 hours, and the last final uh, complete development instar fifth instar is 5 to 6 hours only. So because the silkworm lifespan is very low. So because the continuation process of the calculation of individual methods to one of the larval duration in hours. So how many hours of duration? The good health and good couples are duration of couples very very intense. Now, for example, which is some couples are affected to disease. It is not incubation. It is not the next generation to hibernation and incubation is not performant because the complete good and quality cocoons are duration in hours. Larval duration in hours for to uh, 1 hour to 3 hours. The complete the formation to the next generation. The next one, third one, qualitative characters. Third one, the fifth instar duration in hours. Yes, the very important of fifth instar because the fifth instar secretion of silk fiber. So it is the economical and commercial cultivation. So the both farmers are benefit the 15 stars life stage is important because the 15 star is a secretion to silk fiber. Then it is called as another name is a caterpillar. The caterpillar very well developed to the internal structure and external structure because the mandibles, antennas are very developed. The mandible are functionally it is nutrition. So nutrition. So because this performance is a very important of our life cycle duration of our 15 star. It is called as the caterpillar. So next one, quantitative characteristics is the next one. The preparation the shed structure rates. How 3,000 cocoons couples are silk fiber 5 kilometers like that 4 to 5 kilometers 4 to 5 kilometers one thread fiber. So this is the preparation rate depend by the worms. Then next one I choose the next one, the number of normal eggs in the laying. So the number of normal eggs in the laying, the number of larval egg layings is important because number of normal eggs in the laying because it is the structurally and functionally more than good quality and quantitative eggs are laying by the egg trays, and the next generation produce the next generation of pupation, and increasing the cultures also. So this is the method of process of selection. So completely deserve to the deserve to the four devices like that, individual and mass selections, and fixation of characters, and formation of new breeds, and authorization of new breeds. So individual and mass selections is the appear to the six different methods. So individual and the egg color. The first one, egg color. It is the visualization characterization, and egg chronio colors. The egg chronio colors is the incubation hatching eggs, and next one, cocoon shape. Next one, cocoon shape, and the next one, cocoon color. Then next one, cocoon size, and cocoon nature of grains. So the complete seven individuals methods in visualization and separate to the traits. So because this is the very important. Then next one, mass selections. The mass selection, the internal identification characterization. Internal identification characterization. The quantitative characters. So how to the this scenario phenomenon, the how to the quantitative and characters. The quantitative characters directly indirectly depend by the uh, qualitative and good outcomes are producing. So then this is the mainly uh, abilities in percentage. Ability means is the complete hibernation, hatching, laying. So complete ability is depend by the genotypic of specific species of insects and species of silkworm insects. The next one, larval duration in hours. How many times have you taken the larval duration? Because the larval duration uh, one generation to another generation. How many taken the times of hours, times and hours. So how many taken the hours? Then next one, the fifth install duration in hours. They are very important. See observe the fifth install, because silkworm life cycles are silkworm life cycle 15 stars is very important, because the 15 star is the secretion of silk fiber, because the 15 stars are very important, important to the duration of hours. This is the mainly calculation of hours, mainly calculation of hours in duration of in duration of duration of 15 star. The next one, preparation rate in percentage. Preparation rate in percentage is a very important of preparation rate, because uh, the preparation is the incubation, hatching, the laying of the life cycle of silkworms. The next one, number of normal eggs in the layings. How many number of legs uh, in the uh, number of eggs is functionally formatted uh, to the to the particular this particularly complete number of eggs. How many number of eggs are laying in laying the structurally and functionally. So very important this process. So because each and every one every procedure is very important. This is the selection method of sericulture and cereal. So tomorrow, next class, I will inform the next class. Tomorrow, I will inform you the complete the subject materials of the how to the fixation of characterization, and how many formation of new breeds and authorizations. Okay, thank you one and all. Right.